Good morning, everybody. We are in thematic session day one, where we have, I think, 6 very interesting presentations. First of all, I would like to thank everybody who came here this morning to see these presentations. Of course, the organizing committee for offering us such an incredible conference until now. So, we'll go with the first presentation for this session. Is from co-chair Dr. Elena Bancalari from University of Parma with the title Impedometric Analysis, an alternative way to study different aspects on food microbiology. Elena, you can start.
Thank you for your nice introduction. So good morning, everybody. I'm Elena Bancalari, I'm from Parma, and today I'm going to present the application of impedometric analysis to food microbiology throughout the years. So first of all, we have to define the term impedance, that is the total resistance that occurs in an alternating current circuit. And more in detail is when we have 2 electrodes that are placed in an electrolyte-containing solution and a potential difference is applied, impedometric measurement can be defined as the resistance to the flow of the alternating current that have to pass through the conducting medium. But how can we apply this method to food microbiology? In a very simple way, because it's well known that during bacterial growth, they break down big uncharged molecules into smaller and charged ones, and all these changes together lead to an increase of the total impedance in the growth medium. So then, if we plot this measurement against the incubation time, the microbial growth can be graphically represented, but also measured. Now the question is, how can we use or interpret this kind of data? So the first and the most used until nowadays method to use this data is by fixing an arbitrary threshold line, and when the impedometric curve reaches this threshold, the so-called detection time can be calculated. And this detection time is in a certain way correlated to the definition of the lag phase. So is considering to be inversely related to the sample contamination, but also to the cellular concentration. So here I've listed all the publications that I found in literature from 1925 to 2022, just to give you an example of the potential application of this technique through the years. So the first application were in detecting and enumerating specific microorganisms in specific growth media. Then was also applied to the study of food shelf life, but also then it was applied for the study of microbial metabolism, especially in growth medium, but also in some milk products. And then we arrived to the final and the more recent application of this technique that are in the modeling of the growth of pathogenic bacteria and also to the study of lactic acid bacteria behaviors. Despite all this application, the most used one remain in food hygiene. In fact, for years, scientists have tried to correlate and the impedometric analysis and try to substitute the most common used methods to count bacteria such as plate counts, and that is well known to be time-consuming. But is it really possible to replace plate counts with impedometric analysis? Scientists have tried for years because of its evident advantages, because this technique is very fast, and it gave also information about the physiological state of the microorganism we are analyzing, and it allows the analysis of a lot of samples at the same time. But to correlate impedometric analysis to plate count, a strictly correlation between the detection time and the plate counts have to be reached. And thus, a careful calibration must be achieved. And these need a lot of sample to be analyzed to confirm the data. But despite this, the advantages has prompted several authors through the years to try and look for a correlation between the two techniques. Unfortunately, not all successfully. In fact, 14 out of 29 did not find any type of correlation between the two techniques. And this was because even if small changes in the condition of analysis, such as temperature of incubation, the composition of the growth media, or even more, the concentration of microorganisms, or the physiological state of the cells differs, also the results of the two techniques differs. And this makes the relation between the two very difficult to achieve. And this scenario is getting even more complicated if we think about an application in foods, where a greater variability is apported from the abiotic and biotic components of the food itself. So, for years, scientists have limited the application of this technique to the study of the first part of the data set, and thus the first part of the curve, by analyzing the detection time, as we have already seen, and the lag phase. But from 2016, our group decided to look deeper into this kind of data and try a new way to use it and to interpret it. So it was decided to use and study all the data set and the whole impedometric curve. We have done this by applying a predictive model, and we decided to use an Excel add-in, and through the use of Gompertz equation, we were able to definitely and objectively describe the whole impedometric data set and the whole curve by only 3 parameters. In fact, we obtain the parameter lag, that is well known to be a measure of the time that the cell need to adapt to the growth condition and start their growth. And we also obtain the parameter rate, that is directly correlated with the acidification speed. So we are now able to measure the acidification speed of the microorganism. And last but not the least, epsilon n, which is the maximum value of impedance recorded, and it was associated with the acidifying capability of the strain. And the higher the value, the greater the amount of acid produced by the strain that we are analyzing. So this new approach has turned impedance analysis to a real high-throughput method. And this allows us to apply this method in a very, a lot of ways. So we apply this technique to the study of the acidifying performances of starter lactic acid bacteria in a wide range of variables. And afterwards, we also study the probiotic and non-starter growth behavior, both in the growth media, but also in foods. For example, we use as a substrate different kind of juices or milk products or also sourdough. And then by studying in a different way the impedometric analysis, we were also able to study and predict the isopolysaccharide production by lactic acid bacteria. And finally, we also apply this technique to the study of the effect of natural or synthetic compounds on lactic acid bacteria growth behavior, but also to spoilage and pathogenic bacteria. So, in conclusion, the take-home message is that if the principle of this technique we have seen that is not new, it dates back on 1925, and despite it has been applied for years, only if approached as we have done with a broader vision, this method could find a variety of potential application in food microbiology, and also in this way can be considered a very high throughput method. So I finished my presentation, and I would like to thank the organizing committee for inviting me to present today my work. I also would like to thank all the food microbiology group of the University of Parma, and in particular Professor Erasmo Neviani and Monica Gatti, and of course you for your kind attention.
Thank you for this interesting presentation. Is there any question from the audience? There, I think.
Hi, thank you. Michael Callanan, MTU in Cork. Thank you for a very good and interesting overview. I've worked a bit myself with carbon dioxide and impedance. What you were talking about here, was it just strictly acid detection using impedance, or did you look at other metabolites? Because I know we found quite a bit of variability depending on the organism, depending on the matrix with impedance.
Yeah, thank you for your question. Yes, the first metabolites that we detect with our instrument is acid production. So, because we know that is the first metabolite of lactic acid bacteria, so it's really easy to measure because it's a huge production. But we are also able to measure other metabolites, for example, carbon dioxide that is produced by yeast or other heterofermentative microorganisms by using different cells. So the same instrument, by changing the cells that we use for measurement, we can measure other metabolites that are correlated to the growth of microorganisms. But with this technique, we are also able, as I've already told, the isopolysaccharide production, for example, because on the other hand, they interrupt completely the current transfer. So at one moment, the curve drop. So we are able to measure also this kind of metabolites.
Any other question?
Yes, here on your left, Wellington Pinto from Brazil. Great talk. I got really interested in your work. I wanted to know if you tested like in real foods or was it in broth?
Thank you for your question. Yes, we also perform some analysis in foods. For example, we use some juices, fruit juices, vegetable juices. We have a quite big experience in milk products, so fermented milk. And we also are now working on sourdough, and it's possible if the quantity of water inside the sourdough is enough, we are also able to measure at the same time the growth of bacteria and yeast by using the two cells that I was talking before.
Sure, thank you.
You're welcome.
I think we have time for one question more. I saw there Dr. Mohareb. Fadi, you can... yeah.
Slow down. I was wondering, since you started your conversation or your talk by comparing between the plate count and your method, and you showed the differences and how faster it can potentially be, I was wondering if you tried to develop some mathematical modeling basically to use the impedance as a proxy for plate count. So basically use the measurement to predict lactic acid bacterial count from a plate, for example?
Thank you for your question. No, we haven't did this kind of correlation. We haven't applied any mathematical models because we would like to set up a new approach for this technique that was completely diverse from the one used until nowadays. And so, no. Thank you.
I think it will be a good start to do this.
Okay.
So, any other question? I think we'll thank you again, Dr. Baccalari. So, we'll proceed to the second presentation, which is entitled Shotgun Metagenomics as a New One-Step and Fast Method for Strain-Level Foodborne Outbreak Investigation. It's from Mrs. Florence Beiters. Sorry for the accent.
Hi, everyone, and thank you for coming to my presentation today. Thank you to the organizing committee for letting me present my work. And I will talk about how shotgun metagenomics can be used for strain-level foodborne outbreak investigation. So when we investigate a foodborne outbreak, we try to detect the pathogen that was present in the human samples and in the food samples. And then we characterize them to obtain a fingerprint that we can compare to see if it was exactly the same strain that was present in the human samples and the food samples. And this is done by looking at the genome. But this, called strong evidence outbreak resolution, is only obtained in 15% of the cases nowadays in Europe. And in 85% of the cases, we only get a weak evidence outbreak resolution. And this can be because the bacteria or virus is not fully characterized, maybe because the bacteria was not isolated, or because the pathogen was just not detected in the food. So we cannot say which food caused the foodborne outbreak. This might lead to the outbreak continuing, so more people being infected, or the wrong foodstuff might be incriminated, and this leads to economical losses. So the conventional methods for bacteria, after reception of the food samples in the laboratory, this sample is enriched. We screen to detect the pathogen of interest, and then we try to isolate this pathogen. We type it, and then later on with other methods, we can do a traceback analysis to relate the strain with the human cases. So this takes about 3 weeks, and as I said, it's in 2 steps. So first we'll get the typing information, the serotype, maybe the virulence genes present in the strain, and then later on the relatedness. And now a new method that has emerged is isolate sequencing, and we get the ultimate information because we get the full genome of the pathogenic strain. So we get the serotype, all genes that were present on the genome, and a SNP level relatedness. But it also takes about 3 weeks to obtain this information. And the main limitation is the isolation of the pathogen for bacteria. This isolate, as I said, is not always obtained. It takes time to obtain an isolate, and sometimes there's even more than one pathogenic strain present in a sample. For viruses, it's a bit different. For example, for norovirus or the hepatitis A virus, we receive the food samples in the laboratory, then we extract the RNA, screen for the pathogen, and then we can do a Sanger sequencing to type the virus we have. And there's no traceback analysis. It's only comparing the subtype that was obtained from the food samples and the human samples. Again, sometimes there's more than one virus or one strain present in a sample. So there's a need for a new method that can detect the pathogen in the food samples at very low levels so that it can be implemented in routine, that could detect potentially more than one strain of the pathogen, link the pathogen found in food samples to human cases of the foodborne outbreak, give at least the same information as the current methods, so detect the virulence or AMR genes present on the genome, type the strain. It should also be a fast method and therefore avoid bacterial isolation. So what we present is a shotgun metagenomics method. And for the laboratory part, so we have our food sample, we have the plant cells or the mammal cells, or depending on the food matrix, and then all the bacteria and viruses living on that matrix. And we extract all the DNA or RNA of that sample. We prepare the library, we sequence, and we analyze. The bio-IT data analysis is not so straightforward. That's, I think, the most difficult part in metagenomics, and it's a bit like looking for a needle in a haystack with your computer. You have all these cells from which you have the DNA sequenced, and you just want to characterize one pathogen that was present in all this mix. So we have all the sequence reads, and first of all, we look at the species that these reads belong to. So we do a taxonomic classification, and we try to identify if a pathogen was present in the sample. Then we try to infer the genomes of each strain that was present in the sample. And once we have these genomes of each strain, we can characterize them by doing gene detection to obtain the variance genes, the AMR genes, or the serotype. And we can also do relatedness to link the strain that was found in a food sample to the human cases of the outbreak. It is still a very new approach. So we first developed our method on samples that were artificially contaminated at a known contamination, and we did it with a pathogenic E. coli stack. So we worked with goat cheese, and we spiked it with 2 strains at the same time, 2 different stack strains at a very low level of 5 CFU. Then we enriched for 24 hours in buffer peptone water, and we sequenced on Illumina. So we could obtain, here underlined, the 2 different strains that were spiked in the sample, and we could characterize them. So you can see the green blocks, they represent the genes that we found in the inferred genomes. And we could also relate these strains by phylogeny, so we could cluster the strains from the metagenic samples to the respective isolate that was spiked in the sample. Then we worked on a Salmonella outbreak that occurred in a hotel school in Belgium, and we received from the National Reference Laboratory of Belgium the full meal and the sauce that were linked to the outbreak and positive for Salmonella with qPCR. So these samples were also already enriched by the National Reference Laboratory. We sequenced all reads, all DNA of the sample, all DNA of the sample using Illumina. And first of all, we did taxonomic classification to identify the pathogen that was present in the sample. And you can see here in red that Salmonella represented a high percentage of all the reads that were sequenced from the 2 samples. Then we could infer the strain and type it as Salmonella enterica subspecies Enterica serva enteritidis. So it was very detailed typing. And we could also detect an EMA that was present on the strain genome. And finally, you can see here again underlined the 2 strains that were obtained from the 2 metagenomic samples. And we could cluster them with, in blue, all isolates from food and human origin that were linked to that outbreak. And we could also very nicely separate these strains or this outbreak cluster from sporadic cases in black that occurred in Belgium at the same time period, and from another Salmonella outbreak that was occurring in Europe at the same time period. So in that case, there was only 1 strain that was detected in the sample, and it was fully characterized. Because we could study a foodborne outbreak in real time, at the time, we could compare the timing of the outbreak resolution with our new method. And we could obtain the same result as with a conventional method, so the full characterization of the pathogen and phylogeny, within 1 week. Now, what about long-read and real-time sequencing? We sequenced the 2 previous projects on Illumina, but we can also sequence long reads with Oxford Nanopore technologies or MinION, and it also offers real-time sequencing. So we sequenced a minced beef sample that was also artificially contaminated with STEC, another strain, also at very low level. And the minced beef also presented a non-pathogenic population of E. coli. We enriched for 24 hours, and we sequenced both with Illumina and Oxford Nanopore sequencing. So we could separate the 2 strains, the non-pathogenic strain and the STEC strain, and characterize that STEC strain. And after only 12 hours of MinION sequencing, we were able to obtain the strain, characterize it by detecting the virulence genes that were typical of that strain, but also the serotyping genes. And we could also relate it to the respective isolate that was spiked. Finally, I talked a lot about bacteria. We also tried with viruses, in this case norovirus, that we spiked on raspberries at different concentration levels. For viruses, there is no enrichment possible, so we tested several sample preparations instead to enrich for the viral loads in the sample. And we sequenced with Oxford Nanopore technologies. I will not go in detail, but we were able, depending on the sample preparation and the contamination load, to detect and characterize the virus and to do relatedness of the virus in a phylogenetic tree. We were also able to detect another virus that was present in some of the sample, the hepatitis A virus. This is still a work in progress. It's more challenging than working with enriched bacteria. But so far we could see that depending on the viral load present, targeted enrichment or cDNA random amplification was necessary. And the result also depended on the RNA extraction method. The publication is underway. So, some conclusions. We presented a new method that can detect the pathogenic bacteria in the food samples at very low levels, potentially also viruses. So it can be, in the future, implemented in routine. We could link the pathogen from food samples to human cases of the same outbreak, so it's a strong evidence outbreak resolution. We give more or the same information as the conventional methods, the detection of virulence genes, AMR genes, the serotype. We can potentially characterize more than 1 pathogenic strain in a sample, and we can go faster and avoid the isolation step. So I want to thank everyone who participated in these different projects and made it possible, and thank you for your attention.
Thank you, Ms. Butters, for such an interesting methodology that you present us. We have only for one question there, the lucky one.
Hello, so thank you for the interesting presentation, but I would like to, if you could possibly elaborate a little bit more into the methodology, because at some point you talk about like reconstructing the genome of the strains, but then when you, for example, showed the phylogenetic tree, it was more based on the sample itself. So is it SNP based or is it assembly based, the strain level that you achieved? And second question, does it change somehow the methodology that we apply when you apply the long-read sequencing?
Yes, so I will try to answer quickly, but it's all detailed more in the papers. We first do the taxonomic classification to detect the pathogen that we want to look at, and then we use reference-based assembly, and we use a large database of all the reference sequences of that particular pathogen, and there we can see clusters of reads that we assimilate as the strain. So for example, for the meat sample, the non-pathogenic strain, and then the STEC strain, we could differentiate the reads based on this reference-based assembly. And then we use these reads to do the gene detection and then the phylogeny. And then when we do long-read sequencing, it was basically the same idea, but not the same tools.
Okay, thank you.
So if you have any other questions, I think on coffee break you can find Ms. Butters to... Thank you again.
Thank you.
So the next presenter is Dr. Sabrina Cadel-Six with a presentation titled qPCR method for identifying highly pathogenic Salmonella serotype.
So I would thank the organizer to give me the opportunity to present today the molecular serotyping methods developed in ANSES to identify the most frequently isolated Salmonella serotypes. This work was done with people with expertise in microbiology, molecular biology, and bioinformatics. I think that everybody knows Salmonella, but I want just to remember that in the United States is the leading cause of foodborne illness and the second cause of foodborne illness in Europe. Today, the conventional serotyping methods is used to identify Salmonella. It is a method based on the slide agglutination. This method is long because it takes 4 days for identification, at least. It is expensive because of sera, and it needs animals because of the production of sera. As we saw, several reference laboratories and big laboratories is moving their whole genomic sequencing. But anyway, also these methods is still expensive, can be long, and need particularly bioinformatic skills and equipment. So it is why in ANSES we wanted to develop a PCR method to identify Salmonella serotypes most frequently isolated worldwide. It is known that 30 serotypes dominate agro-food sectors and human infections worldwide. These serotypes account for 76% of human infection in the United States and around 80% of animal, food, and feed samples and human cases recorded by European Food Safety Agency in Europe. Among these serotype 10s, here in red in the list, are considered high pathogenic. It is because these serotypes are multi-drug resistant, they are involved in outbreaks, and they are mandatory in Europe and monitored in the United States. Most of the time, the PCR markers that are commercially available or published in the literature are not specific and sensitive enough to identify these serotypes. We calculate the reliability of 74 published markers used in commercial tests, and no one of them were specific or sensitive enough. This work is published in PLAS, and we realized that it was due because of poor panel of strains used for the validation, and also because of polyphyletic serotypes. We made a deep phylogenetic analysis of more than 600 genomes of Salmonella, and we were able to describe polyphyletic serotypes, what it is. A polyphyletic serotypes include 2 or more genetic lineages. And as we can see here, with the standard conventional methods based on agglutination, these different genetic lineages accounts for the same serotype. And most of the time, only one of these lineages is detected by commercially markers or markers published in the literature. With our assay, PCR assay, we are able to detect polyphyletic serotypes. But here it is the workflow of the development of our Salmonella molecular serotyping method. So we started from analysis of more than 3,000 well-characterized genomes of Salmonella for 70 serotypes to study the population diversities and to implement bioinformatics tools to identify specific and sensitive sequences for each serotype. So we designed primer and probes on these serotypes, these sequences, and we tested them by blast with a local databases of more than 6,000 genomes and 200 serotypes. So the candidate markers were finally validated experimentally by high throughput qPCR, and the final validation was calculated on the basis of results of more than 70,000 PCR reactions. So great advantage of this validated method is the fact that we used high throughput qPCR system based on chip in biomark system. As you can see here, we have a chip. We can load it with a lot of markers and a lot of samples, and all of them cross inside the chips for cross-PCR reaction. We have here the results. In one chip, we can run more than 90,000 PCR reactions. Every point here is a PCR reactions. So you see how powerful this system it is. And just to give you an idea, in the genomic platform of ANSES, we are able to run 1 million PCR data points per year. So now, the success of a PCR method developed is based on 3 assets. First, the phylogenetic analysis that allowed us to identify polyphyletic and different genomic lineages for each serotype. Second, the power of the high throughput qPCR methods used for the experimental validation, and finally, the large panel of strains used for the validation. This slide sums up the panel of strains. So we used 900 strains. As you can see, we have Salmonella enterica, of course, Salmonella other than enterica, but also strains other than Salmonella and 10 surrogate plasmids. So we crossed 77 genetic markers experimentally with all of these strains to generate more than 70,000 PCR data points. And our methods has an agreement of more than 99% with the conventional serotyping methods. So here it is an example of results. We have here the chip. Every point is the PCR reactions. We were testing here different DNA dilutions and replicates. The positive reaction are colored by yellow to red, and black point are PCR with no reactions. And as you can see, the results are excellent. We have the curve on your right, and you can see that we were able to identify some Salmonella serotypes that are really prevalent in the food, animal, human, and feed, such as Typhimurium, but we are also able to identify the monophasic variants of these serotypes that it is now prevalent everywhere all over the world, and also enteritis and others. So we designed and validated several markers for several serotypes, and now we can imagine to design a test specific for each food sectors. And in this way, we can test a great amount of strains. For example, here, we target here serotypes that are prevalent in chicken sectors or turkey, or beef sectors, pork, or feed. And stars indicates multi-drug resistant serotypes, and the highly pathogenic serotypes are underlined. In other way, we can also imagine to design a test specific just for one serotype. And for example, in case of outbreak investigation, check for a large panel of strains and look at the positive results. And we validated this approach with Salmonella bovis morbicans outbreaks that occurred in France with dried pork sausages. So we were able to detect directly in DNA extracts of pre-enrichment broth of sausages the Salmonella bovis morbificans responsible of the outbreaks. And this approach was also very interesting because we were able to identify specifically the epidemic clone sequence type 142 responsible of the outbreak. So Salmonella bovis morbificans is a polyphyletic serotype. So we were able to detect it, identify it really precisely and directly in the pre-enrichment broth. So this kind of approach can be used also with other applications, such as investigation of presence of Salmonella along a factory production chain to make source tracing, to identify the source of contamination, but also to check the efficiency of corrective actions along a factory production chains to eliminate one specific clone or check appropriate cleaning procedure or equipment surface modifications. So the benefits of the PCR molecular serotyping methods are several, are a lot. It is really cheap. It is a PCR reaction. It is fast. As you can see, you can check a lot of samples in a time of one amplification. And we are able to identify polyphyletic serotypes and also strains rough. It means strains that are not agglutinable and not identificated with conventional serotyping methods. And the results are objective, easy to read, and as I tried to show to you, it's really flexible. We can target several prevalent serotypes because we want to investigate special food sectors or just one serotype in case of outbreak or identification of serotypes along food production streams. So we can really design the test on demand following the need. I would like to thank Arnaud Felten and Patrick Fack, who are co-developers of this PCR ANSES assay, Mylan Tran and Vivian Morel, who made part of experimental analysis, but of course all collaborators who applied, supplied or sold us strains for the validation analysis. So here you have the list of the different Salmonella serotypes we are able to identify, and for sure, thank you for your attention, and I will be happy to answer to your questions.
So thank you for your very interesting presentation. So I think we have time just for one question.
Hiya. Thank you very much. Fascinating work, really, and a very impressive diagnostic method. I understand that the bioinformatics database, so the phylogenetic analysis that you perform on different strains is key here to that diagnostic method. And I was wondering if that pipeline is publicly available, so for other research institutes that can use. And how easy do you think it is to adopt the same pipeline for something beyond Salmonella? So while I was following your interesting talk, I was thinking of something like drug-resistant malaria, for example. Can the same pipeline be applied there?
Yes, thank you for your question. So first, I think that the workflow in general it's mean deep phylogenetic analysis of organism, genomic population diversity, so first step, and after that, use of a tool to identify specific and sensitive sequences and in silico validation and experimental validation. So all the workflow can be applied to every pathogens, I mean. Phylogenetic analysis, there are a lot of tools to make it today, and it depends by the availability of a genome. The tools to identify specific and sensitive sequences, there are a lot of tools, free access tools that are available. We used particularly a specific one that was developed in ANSES, but I mean you can find some that are free. And after that, I mean I think that you can use this approach for malaria, for example, as you mentioned. And I also thank you for this question because you point attention on an important aspect of Salmonella classification today, that is nomenclature. I mean, until today we use a serotyping name, but with the advance of the genomic era, we have to adjust this nomenclature and integrate results of genomic. And in this way, we proposed to use the serotyping names, but include also lineages information. And I think that is really important. So we can speak of Derby lineage 2. It's just a little step, a little evolution in nomenclature, but that have a lot of important, that is really important. And I think that is really important also because of characterization of Salmonella harvest. I mean, we were able to show that Derby, that is well known to be prevalent in pork sectors, is characterized by 3 genetic lineages. And finally, 2 of them are prevalent in pork sectors. One of them is most frequently isolated in poultry. So we see that go deeper in genome analysis and implementing in the nomenclature these results, we can really have deeper understood of Salmonella harvest. And to answer better, my colleague from ANSES, Benjamin Felix, that will present after me, will show that we were able to use the workflow also for Listeria monocytogenes. But I don't want to tell more because Benjamin will explain it well.
I think if your team haven't already published the protocol to go from genomes to primer design, I think it should be very beneficial to the research community beyond our food friends here. I think quite a lot of researchers will be interested in that. Thank you very much.
Thank you.
So thank you again to Dr. Kader Six. So the next one is Dr. Dimitra Douro. Sorry for the pronunciation. Sorry. No, sorry. Sorry.
Yeah.
Estelle Shea, sorry.
Don't worry.
With a presentation titled A Text Meaning Approach to Enhance Knowledge of Food Microorganisms.
Hello, everybody. Thank you to the opportunity to let me to present my work with. This work was made by a team of mathematics and informatics at INRAE. And so today I will talk about something different than you listen today. So a text mining approach to enhance knowledge about food microorganisms. So the constat is there are a lot of data, an abundance of data in food microbiology, as illustrated here, in fact, because there are a lot of publications that are published per year, and you can see here an exponential evolution of a scientific publication focused on food microbiology. And food microbiology is also has also an issue to study microbial ecosystem. One of question is which microbes can be live in an environment. So environment could be human, could be a seawater, or obviously food. As presented today, there are a lot of kind of techniques to culture-based methods, so impenetrometry and so on. And at the end, we provide some information about which microbe could be grows in an environment. And then you published this information in databases or in scientific papers. So there are a lot of information, and we can use it. Because cross-referencing information between taxa and habitat is a need to understand microbial diversity, form microbiome, interaction of microbe and ecosystem, phylogeny, and so on. It could be applied in the multiple way to human, plants, animal health, for growth in a specific environment, for a specific use, for example, for depollution and so on, or in case of development of new food, for food processing or food safety, obviously. But there are main problem, in fact, because there are a lot of kind of sort, in fact, of type to express same concept for an habitat, for example. So I illustrate here by cheese, because I'm French, so we love a lot of cheese. And there are a lot of kind of cheese, and there are a lot of expression to speak about cheese, in fact. So we propose a knowledge base to centralize, structure, and standardize the information that's published in some databases and scientific publication, to provide some of this kind of information that could be easy to access and could be reused. This is in the way of the FAIR principle, so find the data could be accessible, interoperable, and reusable. As the mostly of this information is expressed as a free text data, we need to structure them, and if you want to combine them with databases and publication, for example, with other data. To do this, there are different steps. The first one is to detect the information that we need. Then we associate them with a shared reference, so for example, a taxonomy or an ontology. We link them together to make them, at the end, available in the databases. So to do this, we use a text mining method. And we search this information in different sources. So I mentioned scientific literature, so we collect abstracts from PubMed, because in PubMed we have this kind of information. So for example, in this abstract, you have the information about the yeast that could be found in the cider. And we also use some of databases, so biological resource center databases or genetic databases, because this information is also spread. The link between a microbe and their habitat, where they live, is already mentioned by the entry. But, for example, the sources of the sample is mainly in free text, so it's not standardized, so it's not easy to findable and reusable. To do this, we use automatic information extraction to extract information from text. And we base this information extraction on a knowledge model. A knowledge model is the word to just represent the information that we need to extract. So for example, here I represent 3 entities. So I'm interested from microbes, so bacteria, for example. I need to know where this bacteria lives. So there are a relationship with bacteria and any habitat. And for example, I'm also interested by the phenotype that will be expressed. So I have a third entity, phenotype, and a link between the two. We have a text mining pipeline that could be allowed to feed 2 data access systems. So we use documents, as mentioned, databases or scientific publication, with the help of some resources to standardize it, so ontology or taxonomy. Then we are at the step of automatic extraction information. So I could briefly resume it in 3 steps. So there are a first step to detect in text the entity recognition, so which expression of which word is a microbe, which other expression is, for example, a phenotype, and so on. Then there are the second step is to standardize it. So, for example, this word salmonella could be linked by the number equivalent in the taxonomy for the NCBI, for example. And then we extract the relation, so the link between microbe and habitat, microbe and phenotype. At the end, so we propose a lot of kind of relations. Then we integrate it, we sometimes fusion it, and index it for, to feed the information system. There are two. A database named Omnicrob that could be accessible with a web interface, also an API to easy take any information with informatic way, and also a bibliographic search engine. Now some result about this text mining approach. From scientific literature and databases, we can extract a lot of information, around 1 million of link between microbes and habitats. So the most of the information is provided by PubMed information, but also databases add different kind of information. PubMed also provide the information about phenotype. And this kind of information extract could be accessed for you by two ways. So I will be briefly presented here. So the first one is a semantic search engine. So you can flash it, the QR code, or take a photos if you want to taste it. In fact, why is a search engine? Because is a search engine has, as you know, you can search any word, but it's allowed to provide a way to carry a specific link. So I can propose some of query about microbe, for example, lives in habitat. So I take here an example of query. So for example, if I want to search a text with E. coli lives in a meat, for example. And I want also, in the same text, I want the text that explain E. coli exhibit an antimicrobial resistance. So I can type this kind of query. And at the end, I have an abstract. In fact, there are around 20 that could be is the answer of my query. You can see on the right the interpretation of the query by the search engine. If, for example, for the most of you, when I mention E. coli, you think to Escherichia coli. In fact, with the taxonomy, E. coli is also a synonym for a parasite, Entomoeba E. coli. So the search engine understands what is the signification of the E. coli, and explain, in fact, with the highlight where they extract the information in this text. The second access to the data extract by text mining is the Omnicrob databases. So, as you can see here, the URL or the QR code. The main page proposed to see different way to query these databases. So I can search where my microbe live, or if my food, for example, have a specific microbe already found in the literature, and so on. So I propose today just to find this way. Donc I can click here directly on the appel. There are an input file that could be automatically completed. So I take cheese because I'm French. I can also search, in fact, the word used to classify, because it's a classification by a navigation by an ontology. And at the end, there are some of results that could be exportable because it's in table format, so it could be exportable to Excel or other kind of tools to be read. And in this information, in this table, there are a lot of information. By the little plus at the beginning of each line, there are information that could be extracted to understand what the text mining tools do, in fact. So you can see here the real occurrence in text that match with the fresh cheese and how author write it, in fact. And you also go from Omnicrobe databases to Alvis search engine by clicking on the number that are the PMID number linked to the publication. And here you can see where the information that you see on the table is in the context of the text. To conclude some of perspective of this work, we provide we will to add some of new databases as sources, and we also work to improve a text mining approach and the prediction, because as mentioned at the beginning, the text mining needs to predict, to modelize, and obviously there are some of error, and so we improve a lot the new text mining to use the best tool for this subject. For more detail for these two tools and all of the text mining pipeline, you can see these two papers. And I just want to thank Hélène Valentin and Stéphanie Dutch for their help for this project, and more generally our researchers involved in the florilege and Omnicrobe working groups. And I'm available for any question on the subject. Thank you.
So thank you for your presentation, and we have time for one question.
Thank you very much. Very interesting talk. So if I understand correctly, you are using dictionaries and natural language processing for doing your text mining, right?
We use resources as a taxonomy and ontologies, yes, yeah. But also natural language processing, yes, to understand effectively. Yeah. Behind the 3 steps, in fact, there are a lot, around 100 of tools, mm-hmm, to express effectively. There are different ways to arrive this work.
Yeah. I guess natural language processing has some difficulties. It's not very easy to differentiate between E. coli was found and E. coli was not found. So do you have any statistics of the false positives? Have you read any of these abstracts, randomly 500 abstracts, and see what's going on?
Yes, absolutely. In the paper, in fact, the first one, there are an analysis of error, which kind of error the tools done, if they are difficult to find habitat or taxon, or the link between the both, because it's very difficult to detect the link. It's with the help of natural language processing, it could be supported by some of verbs, lives in, and so on, grows. But there are some of a way to express more, less easier to read, and so we have this analysis in the first paper. Yeah.
So what is the false positive rate?
Because we identify, in fact, all the step, the false positive rate. So there are a lot of kind of the. So but I'm happy to follow the paper. Yeah.
Okay. Thank you again to Dr. Shea. Now the next presenter is Dr. Dimitra Duru with a presentation titled Rapid Microbiological Spoilage Assessment of Chilled Chicken Meat Using Non-Invasive Spectroscopic Sensor.
Good morning. I'm Dimitra Duru, and I'm going to present you the work Rapid Microbiological Spoilage Assessment of chilled chicken meat using non-invasive spectroscopic sensors. This presentation will include an introduction about the current food safety management system, the next generation strategies, methods, results, and conclusions. As probably you all know, the current food safety management system is based on screening of a large number of samples for the rapid detection of microbiological quality and safety. And this is, of course, very important for the industry and consumers, for the implementation of corrective actions on time, the inspection of the food supply chain, and the mitigation of food waste. The whole production process is based on the analysis of the end product. The tools that we currently use are sensory analysis, conventional and molecular microbiological analysis, chemical analysis, and mathematical modeling. However, these tools have some limitations, which are that they are time-consuming, they provide retrospective results, they are costly, sometimes they require high-tech technology and thus highly qualified personnel. They are destructive to the tested product, and therefore their application on an outline in the industry is rather limited. The food business operators, authorities, and consumers, however, need results immediately. The proposed alternative that can complement or replace the existing methods for the evaluation of foods is based on non-destructive rapid methods and cloud computing and storage to handle the massive amount of data generated. This strategy is called the process analytical technology, and it uses the following tools, which are sensors, next-generation sequencing, data science, and information communication technology. With all these tools, we are allowed to real-time measure some critical quality parameters. The sensors that we use are on an outline, non-invasive and non-destructive analytical instruments that can be desktop, handheld, or miniaturized, and they are based on spectroscopy and/or image analysis to measure critical quality and safety parameters. Next-generation sequencing, of course, increased our understanding on the microbial behavior on a molecular level. And to date, NGS focuses and it's limited to retrospective identification and traceability and identified food contamination following an outbreak. Data science comes to solve a challenging issue for food safety, which is the handling of the massive amount of data generated with all these analytical and high-throughput platforms. And data analytics, of course, aids in determining which of the parameters, the chemical or the physical or the microbiological parameters are the most critical for spoilage or safety. Vibrational and fluorescence spectroscopy may provide useful information on the metabolic fingerprint of a product, and in conjunction with machine learning and computational analysis, it may provide information related to safety and quality. The advantages of using spectroscopic sensors are that they require minimal sample preparation. They are, as already said, non-destructive to the tested products. They are very easily implemented in the industry on real time. They are cost-effective. They provide rapid data acquisition and thus estimation of quality. They are environmentally friendly and economic. So we are moving to the research objectives, which was to evaluate the potential of Fourier transform infrared and fluorescence spectroscopy in tandem with different machine learning algorithms and multivariate data analysis to quantitatively assess chilled chicken spoilage. Very briefly, we obtained breast fillets from a local industry. They were stored at 4 Celsius degrees at aerobic conditions in the original package, and they periodically sampled throughout storage. Data were collected with conventional microbiological analysis, FTIR analysis, and fresh detect analysis. We enumerated the total viable counts, the microbial population's total viable counts, Pseudomonas, Brochothrix thermosphacta, lactic acid bacteria, Enterobacteriaceae, and we also enumerated the luminous bacteria, including Photobacterium sp, which has been very recently associated through next-generation sequencing analysis with meats and chicken meat in particularly. The instruments we used was FTIR and fresh detect, a vibrational spectroscopic sensor and fluorescence sensor respectively. And we then correlated spectral data and microbiological data for model development and validation for the estimation of the different microbial groups. Spectral data were normalized with standard normal variation before analysis. Two machine learning algorithms were applied, the partial least square regression and support vector machines. Our models were externally validated and their performance was estimated by calculating different metrics. And in this figure, we can see the microbial evolution during storage of the main spoilage microbial groups. As expected, Pseudomonas were the dominant flora with the orange line, and Brochothrix thermosphacta followed very closely. Lactic acid bacteria and Enterobacteriaceae remained at lower levels. We were also able to count luminous bacteria on chicken meat that were scarcely found at the beginning of storage and was detected in samples from 1 out of 3 batches. But at the end of storage, they were found at populations of up to 7 log CFU per gram, which is considered as the limit for microbiological spoilage. For more information, you can refer to this poster. Here we can observe 2 representative spectra from the 2 sensors at the beginning and end of storage from a fresh and spoiled sample with blue and red lines. And we are going now to the results with FTIR analysis and PLS models that were generated. We obtained a good relationship between the measured via the microbiological analysis and the estimated microbial populations. More than 80% of predictions for total viable counts and Brochothrix thermosphacta, and around 70% for Pseudomonas were included with the plus-minus log unit area, which is considered microbiologically acceptable. Of course, in the case of luminous bacteria, here the results were not satisfactory as expected because their behavior was completely different between the different batches. Again, with FTIR analysis and support vector machines, predictions and observation presented a good distribution around the line of equity. And for all microbial groups, more than 80% of predictions were considered as accurate. The performance metrics of the 2 models generated with the FTIR spectral data were calculated, and square R and root mean square error indicated good model performances. However, support vector machine models perform slightly more satisfactorily than PLS models. In the case of fresh detect sensor, PLS models achieved poor prediction performance. There was no satisfactory relationship between the measured and the estimated microbial populations. The same observation was made when a support vector machine algorithm was used for model generation. It also achieved a poor prediction performance. To conclude, spectroscopic data in tandem with the appropriate machine learning approach presents a considerable potential for the fast and accurate assessment of chicken meat quality. However, this potential appears to be sensor-specific. Under the conditions of this study, FTIR-based models exhibited the highest performance, and more investigation on the machine learning approach is needed to evaluate the suitability of this handheld fluorescence sensor to accurately predict breast fillets spoilage. Thank you very much for your attention.
So thank you for the very interesting speech. So we have time for one question, I think. Is there any questions?
Good.
Good. It was everything. Oh, no, we have one.
Oh, okay.
Thank you, Dimitra. While you were going through your presentation, I was asking Professor Mikas about the luminous bacteria. I'm not a microbiologist, so he was explaining what it is to me. And I was wondering whether the nature of that bacteria is interfering with the hardware, the platform, which is measuring the reflectance. Do you think that's the reason why you didn't get a good correlation there? Or good prediction, rather?
I don't think so. Their growth pattern was completely different between the different batches. That's why we couldn't make a correlation with the spectral data we obtained.
Okay. Good result on all the other bacteria, though. Thank you.
Thank you.
So thank you again to Dr. Duru.
And last but not least, we will have Mr. Benjamin Felix, who he will present us Listeria monocytogenes identification by high throughput real-time PCR by 30 clonal complexes prevalent circulating in food, animal, environment, and human Europe.
Hello, everyone. So I would like to thank first the Congress Committee to let me make this presentation. So this presentation deals with a new method developed for rapid detection, identification of Listeria monocytogenes clonal complexes. It is part of the work program of the European Union Reference Laboratory for Listeria monocytogenes. So a few words about the context. I don't want to go too much into detail into the MLST method, multi-locus sequence typing, because it's now very well known. It's applied on almost all the pathogens. This method now is admitted as a reference to describe the phylogenetic genetic diversity of the pathogens, and is based on two levels of nomenclature. The first level is the clonal complex. So CC, I don't know how to point it, and the sequence type, ST. So the sequence type are a unique code that provide a combination of alleles of MLST that creates a subgroup in the population, and the clonal complex is a group of sequence type gathered into a family of sequence type. You can see in the tree on the left side the phylogenetic structure of the Listeria population according to MLST nomenclature. So basically there is two lineages, lineage 1 and 2, that are the major lineages circulating in food and human cases. And you can see on the right side a zoom on the lineage 1 and the lineage 2. So the big interest of MLST, and I will do it short, is that it creates a nomenclature that is universal. It's a common language to speak about Listeria. One strain, CC1, in US and in Europe, basically have the same characteristics. It's also a good way, it's a way used now as a routine for outbreak description. For example, CC6 was, a strain from CC6 was the source of an outbreak in Europe, a multi-country outbreak. Now it's possible to connect new identification of strain to this outbreak because of this nomenclature. But the drawback of MLST is that you have to sequence the genome, the whole genome of the strain to get it. And it takes between 3 to 5 days and approximately 100 euros, 150 euros per strain. So our project aimed to provide a method that will be rapid and cheap to provide the major circulating clonal complexes in the food sector. So what we did first, we did a maximum of collaboration to generate data. We needed genomes on the strain circulating in Europe, in the food and the food production sector. So we collaborated with, for example, for the sequencing of a lot of strain from France, circulating in food and human cases. We also worked on European project like LISEC. It's a project that provided thousands of genomes on the strain circulating in all European countries. We also work with some of the laboratories of our network. For example, the Slovakian laboratory would describe the diversity based on MLST of their strains during 10 years. We work also with the same kind of study with Poland, with all the maximum of laboratory of our network. We also work on the diversity of Listeria at the farm. For example, cattle abortion diversity of Listeria in Latvia, in Slovenia. And finally, all the laboratories of our network gather their data to create a large amount of large database that provide the diversity of Listeria circulating specifically in Europe. The aim was to be as representative as possible of what is circulating, whatever the region. We also finally work with 7 national reference laboratories of our network and also 14 institutes in Europe to publish this data set of nearly 1,500 strains genomes from food for ALF and for animals and environmental isolated Listeria. So you can see just on the right side, A and B, it's a geographical diversity, A, and according to the timeline, and also the food diversity according to the timeline of the dataset. So all the strains of the dataset were sequenced, and the genome are public. Same for C and D, describing the food from environment, and also the type of environment. So it's a bit small, but there is farm, soil, wild animals, basically. Based on this large dataset, we started when we defined which clonal complexes should be targeted by the method, because we cannot target all the clonal complexes. There is nearly 200 at the moment. So we decided to target 30 clonal complexes, which are representative of 90 to 95% of the circulating Listeria in food. So it's based on the literature we produced previously. And to target these clonal complexes, we designed 40 primers and probes. Our design, I don't go too much into detail, but we use a pipeline that enables to design really specific and sensitive primers and probes based on the genome of the bacteria. So the dataset used was from public databases and also from farmer studies, and is really representative of the Listeria, but not only in Europe. The dataset of genome was representative of the Listeria circulating in the world. So we have strain from 18 EU countries and strain from 19 non-EU countries for the design. So we are not going to design for Europe only, but for a method that could be applied worldwide. but the targeted CC are for Europe. So this method was developed for high throughput system, as described Sabrina Kadelesix before me, but also was validated as a multiplex method, as 10 triplex and 3 duplex and 3 simplex, to be applicable worldwide in all kinds of real-time PCR machines. So just a list of the clonal complex we are targeting. There is 30 real-time PCR based on unique sequence on genes. 4 are on intergenic species. And we completed the panel by an existing method providing the molecular serotyping. That is somehow a double check for the data obtained with the PCR. Regarding the validation, we gather a large data set from 21 EU countries. This trend, it was nearly 600 strains in data sets. This graph presents the different clonal complexes and sequence type used for the validation. In green, you can see the strain identified by the method. In red, the strain not identified by the method. And if we summarize this data, we obtain a specificity that is really good, of 99%, more than 99% for all the couples of primers and probes. And for 3 of them, for the other primers and probes, we had the sensitivity. The sensitivity basically provides the false negative results that are the most annoying issue. And for all the PCR, we did not detect false negative, except for 3, CC9, CC1, 193, and 204. For those one, we tried to design as best as possible, but because the false negative was pretty rare, we decided to maintain a single PCR for these strains. We also highlight that because it could be need to be underlined. There is some cross reaction, some false positive results, like when some strain could be identified by a PCR, even if they are not from the targeted CC. It is possible, but all the CC that are detected by this cross reaction are pretty rare in Europe, and could be even useful when you are looking for such rare ST. I do not go too much into detail for this cross reaction. Finally, we define one panel for the validation, but also one panel to evaluate the performance of the method. So for this, we created a panel of 600 strains, nearly, and these strains are isolated from different food processing environments in France and Europe. And also a panel, as you can see, from bovine. And you can see in column, in line, all the groups defined by the method. For example, you can see that for example, CC1 here is divided into 2 subgroups. Same for CC37, CC3121. It was just to be more discriminant. Otherwise, you can see all the different groups we generate with the PCR. I will not describe the whole table. The dataset is a large dataset from food, as I told you, from different food sectors at risk for listeria. It is exclusively from food processing environment because the main issue in listeria is really the contamination of the factories. And we also added an additional dataset from ruminants just to see the performance of the method on strain from primary production. If we consider the primary production, 78% of the strain were identified by the method. If we consider the strain from food processing environments, we cover 94% as expected, so it's a really large coverage. Considering the strains that are not identified for the 2 datasets, So these strains were, of course, sequenced, checked for the MLST to make sure that there is no issue with the method. And actually, all the clonal complex or targeted clonal complex found by sequencing are not into the targeted ones. Regarding the follow-up, at the moment we are working on the methods improvement, not only to identify strain clonal complexes, but also to identify directly the clonal complex inside the matrix. So we are working on several matrices. We have several projects on this topic. So it can be soil, milk filter, food, swab, animal sample, everything useful for Listeria, and we are going to move on that. The basic target is to apply the PCR directly on the samples to be able to find in one way through DNA extraction everything dead and alive in the sample, and the other way through enrichment medium. So for Listeria it is Alph-Fraser and Fraser to find the strains that are viable. At the moment we did some tests with the enrichment, and we are able to detect 10, even if we had 30 different Listeria in the same food matrix, we could detect it. So it is really interesting, especially to answer the question of the multi-contamination of the food products. As you have seen before, there is several factories carrying a lot of different Listeria, and the multi-contamination is at the moment difficult to assess. And it's also a way to make a lot of sampling for cheap price from the factories and maybe identify better the contaminations. So just to finish this presentation, this method is discriminant, more discriminant of all PCR developed on Listeria, rapid, cost-effective, informative, broadly accessible because you just need a real-time PCR machine. It's available everywhere. It's also a frontline method for the screening to save time and money before to make the sequencing of the strains. It is scalable because we can design really quickly. We need just 1 week to design a new PCR and to validate it with high-throughput machine. It's also open and will be published soon. It will be used in ANSES and also for the network of laboratories that cannot perform sequencing as routine. For routine surveillance, outbreak investigations, it will provide a real tool, really efficient for the laboratories to answer the question asked by the authorities. It will be a way also to monitor the emergence by strains not detected by the method and sequenced after that. It will be also useful for our private partners to make routine analysis in factories for cheap price with a high throughput analysis, and finally provide guidance to improve the hygiene in the food factory environment. I would like to thank all the people from ANSES involved in this project. I would like also to thank the funding. A major part of funding was provided by the European Union Reference Laboratory for the network of labs, and also France Filière Pêche, which is the French Institute for Fisheries, that provided part of the national funding for this method. Thank you.
Thank you very much, Mr. Felix, for your presentation. We have a one quick question time before coffee break. Anyone? I cannot see anyone. So if you have a question, you can join the coffee break with Mr. Felix. And thank you very much for joining this session.